Thank you for the kind invitation, and I apologize for not being present in person at this time. In this presentation, I'm going to cover the concept of AI segmentations for brain gliomas and the volume extractions and what we are using these volumes for. So before we even start, what is the tumor segmentation? Tumor segmentation is a classification task where we have to classify each voxel of the image as involved by the tumor or not. So for each voxel, we say tumor yes, tumor no, essentially. In the most typical implementation, it is just a little bit more complicated than that. Not only we usually say tumor yes or no, but we want to say if tumor yes, what component of the tumor that voxel represents, the contrast-enhancing tumor, the non-enhancing T2 hyperintense surrounding part, or the cystic necrotic portion. So there is a subclassification task, let's say. Of course, the most ancient approach to tumor segmentation is manual segmentation. So essentially just painting on the image where the tumor, where we think that the tumor is, essentially. Of course, this is very time consuming, it requires training, and it is to some extent operator dependent. So one first improvement in this field was represented by semi-manual segmentations that combine some automated components and some manual components. Therefore, it's called semi-manual or even semi-automated. One example that we've been using in the lab for quite a long time is this workflow, for instance, where we have a T1 subtraction map that is calculated by normalizing and co-registering T1 pre and post contrast. So this gives us a semi-quantitative estimate of which voxels have an objective contrast enhancement. Then there is a manual component where we outline the approximate region of the tumor, so it's much faster and than the fully manual segmentation. And then we use this map to threshold and select a cutoff of quantitative enhancement to only isolate the components that are enhancing. Of course, this is a bit more reliable than fully manual segmentations and even faster. Another example of semi-automated segmentation is represented, for instance, by what you can do with ITK's map, but there is a bunch of different software products that can help you with this. The way this works is you select a bounding box where you see that the lesion is contained. You select certain thresholds of signal intensity that you think that your tumor is representing, of course, because in T2 flare, for instance, the tumor is brighter than the rest of the brain. This is the assumption. You also have to select bubbles, they're called, so the seed points where the segmentation is started, and then the software will expand those bubbles to create the final segmentation by respecting the bounding box and the intensity thresholds that you applied. So this is, for instance, one of the results that you can obtain. Ideally, though, we should get to a point where segmentations are fully automated and there is no human interventions. That would be ideal. From a historical perspective, a big step in this direction was represented by the curation of the BRATS datasets starting in 2012, which were publicly available segmentations with images of gliomas that were also expert annotated. So they could be used to train models to do the task automatically. And the first models were more like machine learning models, like random forest, logistic regression, clustering algorithms, and whatnot. In 2015, UNets, so some type of neural networks, deep learning, were started to be used as tools for segmentations of cells in microscopy. And only after that, around 2016, 2017, the same models, because they worked satisfactorily, apparently, were also used for tumor segmentation, and this essentially gave birth to the whole field of deep learning segmentation that constitutes the main automated segmentation method nowadays. One good example of these tools that are fully automated is HDGlio, which is publicly available for free. You can download it and use it. It just requires a little bit of processing. Pretty much what this algorithm does is it starts from 4 conventional sequences and creates contours automatically of the enhancing component and the surrounding T2 hyperintense portion of the tumor. Another tool is Neosoma, which instead is a commercial software that we use a lot in the lab because we have a collaboration with this company that creates a similar automated mask. It also has a necrosis cystic label on top of it. If we want to give a non-exhaustive overview of some tools that are available, I separated the upper row with clinical PACS integrated products from the bottom row that has more research oriented that are not really PACS integrated software products. And then the 2 columns separate semi-automatic, semi-manual, semi-automated solutions on the left. And then on the right, the fully automated AI-based solution. So there are a bit of kind of different combinations. Typically, it's not a strict rule, but usually the PACS integrated clinical products work with a separate workstation, so they are usually connected to PACS. They can send data back and forth with PACS, and they are on a different local workstation. Whereas the fully automated AI tools are usually cloud-based, so usually the data is pushed from PACS to the cloud. The company processes the data, creates the segmentation that gets pushed back to the local computer, essentially. One special mention to brain mets segmentations because they add 1 layer of extra complexity. In brain metastases, there are usually multiple metastases, and the complexity is that the automated algorithm should also account for multiple time points and try to link the same metastasis over different time points to track, for instance, graphs of volume changes of each metastasis over time. Neosoma has a tool for brain mets. RoboVision also has a tool, so you see how each lesion is given a certain ID, and you can track the ID volume over time. And as a separate note, I've been also working on lymphomas lately, and I have been using a glioma model to segment also lymphomas, and it works pretty decently in my own expertise. So maybe that's a tool that can be used. If we want to make a final point, a couple of points about the status of automated segmentations right now, there are open source tools that are free and they exist, but they're not very clinical practice friendly. They require some processing on the back end, and you need some neuroimage processing expertise to use them. Whereas commercial solutions are more clinically oriented, more clinician friendly, but they require payment, so they need an expense justification. PACS integration of the commercial solutions, it depends. Sometimes it's better, sometimes it's worse, sometimes it's a bit cumbersome. And the other aspect to consider is that cloud-based solutions sometimes conflict with institutional policies. So some hospitals are not super happy about sending images and data to cloud to the companies, but that depends on the hospital policies. And one last point that I haven't mentioned yet, it would be good to have the possibility of applying manual edits if you see that there is something imprecise about the segmentation. I think not all tools allow for that. Some of them, they output a segmentation and that's it. Not always there is a possibility of fine-tuning and refinement. That would be good to have. So it's something that if you want to get a tool, you probably should ask if there is a possibility or not. So currently, overall, it's not super easy to quickly implement automated segmentation in the clinical practice, but I do think that the field is advancing very fast, and potentially some of these problems are going to be overcome sooner than later. So now we just discussed how to obtain tumor segmentation. So now the question is, okay, but why do we need them? What do we use them for? As you may know, Rano 2.0 allows the use of tumor segmentation to track the size of the tumor over time and therefore decide whether the tumor is progressing or responding to a certain treatment. One important caveat to understand, though, is that you should still define verify that there is measurable disease, which is in Rano 2.0 in 3D imaging, one by one, a nodule that measures 1 by 1 centimeters in the three-dimensional space, before you use the segmentation. So for instance, in this case, there is measurable disease, segmentation 20.7 cc, you can use the volume for Rano purposes. In this other case, though, there is no measurable disease. You cannot find the nodule of 1 by 1. In that case, the segmentation is not used. You can just use 0. You can abstract the volume to 0. The concept is that we don't want to track the volumes of pericavitary enhancement, linear enhancement, situations where you don't really have an actual nodule of disease. So the point is, possibly this is the one leftover manual task in Rano in case we use automated segmentation. So also to address that, we recently developed a possible software prototype that can possibly in the future help with that. It's still a prototype, but we're trying to have a software that automatically tells you whether there is measurable disease in a segmentation and also outputs automated diameters that are Rano compliant. So as you can notice, they don't cross the cavity, the surgical cavity, but they respect only the solid tissue. So this may be something that helps in the future. Okay, so about Rano, of course, this is not a Rano class because there are other presentations about Rano, but just a couple of points to refresh. As you know, there is a certain amount of tumor shrinkage or tumor growth to consider a time point as a response or a progression, which is 65% shrinkage response, 40% progression, 40% growth is a progression. But it's important to understand 50%, 65%, 40% compared to what? There is a specific reference scan to observe. For response, it is always the baseline. So compared to baseline, you need a certain shrinkage, like in this case, there is a shrinkage compared to baseline maintained for at least 1 month. That is a partial response. But for progression, we don't compare to baseline necessarily, we compare to nadir. So this is the smallest volume of the time series. So for instance, in this case, the tumor grew compared to the time point when it was at its smallest. So the tumor is growing back and this is a progression. Note that this time point with progression, it's not much larger than the baseline. But indeed, we are not comparing to baseline, we're comparing to nadir to assess that there is a progression. So always remember to search for the nadir so in your time series so you can actually see if the tumor is growing compared to the nadir. For confirmation scans, response always requires confirmation scans. You have to observe response in 2 scans for at least a month to call it a Rano response. For progression, you may or may not need a confirmation of progression, which means you see a progression and you want to see that there is further progression, to call it a definitive Rano progression. It depends on the timing, depends on the therapeutic agent, depends on the trial, it depends on a bunch of things. But it's important, if you have to apply the confirmation of progression, that you know how to apply it. Sometimes the common misconception is that if a tumor grows, like here, and then it remains around that size, that the confirmation is confirmed. That's not how it works in Rano. The confirmation of progression works that you have to observe a preliminary progression, so an initial growth, and then further growth. So 2 progressions in a row, let's just say, to call it a confirmation of progression. If after preliminary progression, the tumor kind of stabilizes or even grows insufficiently, that's a pseudo-progression, PSP. And these are 2 examples. And again, you don't have to always use confirmation of progressions. There are certain time frames and criteria, but if you have to, this is the set of rules to apply. I do want to remind that Rano is not only about images, you should also account for neurological and clinical worsening if that happens. And you have to account for corticosteroid doses. We've been knowing from the 80s, essentially, that corticosteroid administration causes a shrinkage in the tumor, especially in the contrast-enhancing tumor, and this also is it's a good example, that is essentially considerable a pseudo response. So when we consider if a tumor is showing a response on imaging or a progression, we have to see if that patient perhaps received more steroids and that's a shrinkage due to steroids, or they decreased the patient's steroids and maybe we see a false progression that is due to a steroid decrease. The whole set of Rano rules is a bit complicated, and also this is a space where we work. We've been working a lot, and we developed a prototype app that acts as a calculator. So you could input all of the measurement in different time points. It will output a plot, and it will output what time point is a response, what time point is a progression. This is at the moment implemented for modified Rano, not so mRano, not for Rano 2, but they're kind of similar. And we'll see how it will be distributed. So maybe that's something that helps in the future. One potential objection is that do we really need volumes? Because we just reviewed the Rano rules, but the Rano rules also apply to diameters. We've always done measurements with diameters. Why do we need volumes? So a very compelling situation that I like to show is low-grade gliomas that grow very slowly. I want to say that here it's very complicated to see that this tumor is growing. If I was drawing diameters, probably I would be imprecise enough that I do not, that my imprecisions in the diameters outweighs the actual true growth of this tumor. But with automated segmentations, we can see that it grows very, very slowly. Now, you can make the point that this growth is not clinically relevant because it's too little. That's debatable, but that's a clinical question. It is still important to know that this tumor is actually slowly growing. And importantly, this is not operator dependent because it's done by the AI model in a reliable way. There is literature that shows, if we compare, that diameters are have less interreader agreement than volumes from segmentations, and that they are more subject to this yo-yo effect because they go up and down a little bit more because they're more noisy. And importantly, the interreader agreement was better with volumes in this study, even though it was a semi-automatic volumetric segmentation. So I would argue that when we go full automatic with AI solutions, we don't have any issues with interreader agreement anymore. We talked about low grades, but what about high grades? So a very compelling case, in my opinion, is high grades that have very irregular margins. They're very large. They have very odd shapes. So it's very hard to measure them. If we were to draw diameters in these cases, it's not clear where we should draw diameters. These blue diameters are comprising part of the surgical cavity, so not ideal. The red one and yellow ones, it looks like it's a multifocal tumor, but it's not, so they're not really right either. Other diameters wouldn't comprise the necrosis, or they would just simply underestimate the total size of the tumor. So definitely easier to have an automated segmentation that tells you enhancing volume 86 cc and so on, and it's reproducible and consistent. So definitely a good scenario to use volumes, in my opinion. The debate on whether diameters or volumes should be used to measure tumors has been going on for ages. But essentially, long story short, my point is that yes, they are highly correlated, so it's difficult to see a differential advantage of one over the other, even though there is some evidence that volumes may be better. But definitely, volumes are better in terms of interreader agreement, even when using semi-automated volumes. So let alone automatic volumes really will erase the problem of interreader agreement completely. And I think overall we can make the point that volumetric measurement are equal or better than diameter and should be preferred if available. Hopefully they will be more and more available. Importantly, it's important to know about this study when we talk about follow-up with AI segmentations. They analyzed clinical trial data sets, and they showed a very good agreement of the HD glio automated segmentation with the ground truth segmentation of the radiologist, very good agreement in PFS, whereas the diameter-based radiologist assessments has had some disagreement, and they also further validated the segmentation and time to progression based on segmentation with overall survival. So very strong article in this direction. Um, I want to mention, to step aside from Rano for a second and mention what the tumor growth rate paradigm is. Whereas Rano just compares this 40% growth, 60% shrinkage between two time points, the TGR, tumor growth rate paradigm, the thought is we want to fit the trajectory of growth before a certain treatment and then see how that trajectory changes when a treatment is started. So for instance, here with IDH inhibitors, we can draw a line before the treatment and then after. You can see that the slope of the growth has been reduced, which probably symbolizes that the treatment is working. It's another case where the slope is reduced. These is our own cases where we show that after IDH inhibition, the slope of the tumor growth bends to the other side, or in this case, it's a non-responder, so it did not bend. Actually, it increased, and you can see how the change in growth rate before and after the treatment is associated with patient outcomes, even after adjusting for confounding factors. Of course, you could make the point that sometimes, that often tumors don't behave with the linear growth, so you can even use bi-exponential modeling and draw and pull some statistical parameters from the exponential modeling and show how these parameters you estimate with with mathematical modeling are associated with patient outcomes is another example that I used by exponential modeling for. And then I want to mention this very great article where they used an even more sophisticated statistical model to account for all the volumetric changes of all patients in the same model. It's called the joint modeling, and see how it was associated with patient outcomes such as overall survival. And they could very robustly show that the growth rate of the tumors with this very refined method were strictly associated with overall survival and time to next intervention in low-grade gliomas. Finally, no, not finally, but just one slice, one slide that I want to show to remind that of course, we spoke about tumor volumes extensively, but we also use tumor segmentations for multiple other purposes, and specifically, we oftentimes extract data from segmentation. So we extract histograms of perfusion, we extract diffusion values and histograms of diffusion, for instance. Any advanced imaging metrics that you can think of, segmentations are useful for that. Radiomic studies and textured features and so on. So implementing tumor segmentations goes beyond volumes and has other applications. And as a final application, I want to mention statistical parametric mapping, which is also an application of segmentations. The idea is that when we obtain segmentations, we can map the segmentations onto a common space and repeat this for every patient. So we have all the patient segmentations stacked in the same common space. And what's interesting is that at this point, all the tumors are aligned in the same common space. So for each voxel, we know patient 1 has tumor in that voxel, yes or no. Patient 2, yes or no. Patient 3, and so on. And we also have other variables from those patients. For instance, the presence of a certain genetic mutation in the tumor. So what we can do is we can conduct statistical tests at the voxel-wise level, calculate p-values on other statistical metrics, and then map them back onto the tumor location. So for instance, this can be used to see if where are the regions with statistically different occurrence of MGMT methylation, IDH mutation, and so on. So statistics at the voxel-wise level. I've recently used this approach for survival analysis, and you can construct a model and use it. Of course, you need quite a lot of data. This was a data set of 600 patients almost. And you can see how you can use the model to say that certain locations of brain tumors in the brain, deep-seated, eloquent brain, are worse for survival, whereas other locations are favorable for survival. You can complicate this further using interaction variables and map, for instance, how a prognostic variable has a different impact on survival in different brain locations. For instance, tumor volume, of course, is associated with worse survival, but in certain brain areas is associated more strongly with survival and impacts survival much more strongly. That being said, this is the end of my talk. Thank you for your attention. Unfortunately, I am not present to take questions, but please, this is my contact information if you want to reach out with any questions or comments. Thank you again.